Hey, welcome back, biology students. You have a test tomorrow. Are you ready? Let's find out. So first, I would be very familiar with the order of events, the list of the phases of the cell cycle. So when it says cell cycle, right, it's including interphase, mitosis, and cytokinesis. Okay? So remember, interphase is G1, S, and G2. G stands for gap or growth. Mitosis, remember PMAT, prophase, metaphase, anaphase, telophase. And it's followed by cytokinesis, or it kind of happens at the same time as telophase. So be familiar with the order of events as well as what happens in each of those events. Okay. You need to know the relative time cells spend in each phase of their cell cycle. So relative means not an exact amount, right? It would be like the majority of the time is spent in interphase, which we can see from this pie chart. Within mitosis, you could say the majority of the time is spent in prophase. All right, so that's relative time, not an exact number, but in relation to each other. So depending on the resource you use, you might find anywhere from 50% to 90%. It also depends on the cell, right? So the type of cell and the nutrients that are available, the environment, all will alter that amount of time that it spends in interphase. You will be given some numbers where you have to calculate the amount of time a cell spends in interphase. And that is totally reasonable because we've done it 3 times in class, right? So that's a skill we should be able to perform. All right, so relatively speaking, interphase is the longest, prophase is the next longest, and typically metaphase or anaphase is the shortest. You also need to be able to describe the events of mitosis, and these are all multiple choice questions or true false questions, so it's not like you have to write them out when I say describe, but you need to recognize description. So there's several questions. You might have a picture, recognize that as we'll just say prophase because we didn't really do pro-metaphase. Recognize that as anaphase, cytokinesis. Okay, these pictures look similar to the jigsaw quiz I think that we took. You should also be familiar with what's going on, not just what it looks like. So if the question were, in which phase do spindle fibers emerge? You would say prophase. In which stage do chromosomes line up at the equator? Metaphase, okay? So you need to be familiar with the major events that are happening. Anaphase, sister chromatids are pulled apart. Telophase, everything is opposite of prophase, right? So the nuclear membrane comes back, spindle fibers disappear, chromosomes uncoil, go back into that chromatin, thin thread-like version, and then in cytokinesis, the cell splits in 2. So we also talked about checkpoints that are like the quality controls. You need to know what those checkpoints are, why they're important, right? So the G1 checkpoint, checking to see if the environment is favorable for cell division. Is there enough nutrients? Is the cell big enough? So this is basically, can DNA synthesis begin? It's easy to remember. The checkpoint is at the end of a phase, right? So whatever comes next, the checkpoint is basically saying whether or not that can happen. So the G1 checkpoint is at the end of G1. So if DNA is intact and all things are good, DNA replication can occur. DNA synthesis occurs during the S phase. Remember, S is for synthesis. That means to build. So the S checkpoint here just before G2, it's going to check and make sure all of that replication occurred correctly and that the DNA is in good shape. So once you've passed the S phase, you are committed to mitosis. The M checkpoint is at metaphase of mitosis. So it makes sure that all of the chromosomes are attached to the spindle fiber and lined up correctly at the equator, indicating that it's okay for the cell to divide. What runs these checkpoints are the cyclin CDK proteins. So remember, cyclin is named because it cycles. It's high at certain points and then it's low. So we can see it's high at the checkpoint, right? CDK, cyclin-dependent kinase. Kinases, remember, phosphorylate things, and when they phosphorylate them, they either activate or inactivate them. In this case, they're activating them. So here you can see the cyclin binding the CDK. You'll remember this from a POGL. And then the phosphates are added when the two come together. So now you have that maturation factor. So that turns, it's like turning the light switch on, which allows the cell to proceed to the next checkpoint or through the next phase. So the cyclin CDK complex is like an on-off switch for your checkpoints. So if they come together, phosphorylation occurs, and that allows the cell to proceed to the next phase. So how is the cell cycle related to cancer? Remember, cancer mostly affects interphase, right? It speeds cell division, so you have a much more rapid division and a shorter cell cycle. We compared the relative percentages, and we saw that the biggest change was in interphase. So remember, interphase is really important because that's when the DNA is checked. So you're kind of proceeding through the cell cycle without a DNA check. So there can be damage to the DNA, which causes cells to be irregular, which leads to your cancer. So there's a loss of quality control. You also read about proto-oncogenes and tumor repressor genes. So those are genes that can play a role in cancer. Proto-oncogenes are supposed to allow for the cell cycle to continue. So if there's a damage in that, it's going to continue too often and when it shouldn't. The tumor suppressor is supposed to kick a cell out of the cell cycle into G0 phase if the DNA is damaged. So if the tumor suppressor gene is damaged, then, or if there's a mutation, then this kick out to G0 and apoptosis does not occur. And again, a cell that should not be going through the cell cycle continues to go through. Comparing cytokinesis in plants and animal cells. So remember, the animal cell is real simple and easy. You got this cleavage furrow occurring, basically just pinching the cell in half, whereas the plants have a little more complex because they have to make that cell wall. So first they lay down a cell plate, vacuoles all line up, they release their cellulose on either side, and that forms the cell wall on either side of that cell plate. So a little bit more work to be done for the plant cells than the animal cells. Okay, number 8 on your review just says to be able to interpret data in graphs. So this is really just graph reading skills. All the information is there for you. You might see a pie chart. This pie chart uses percentages. I believe the pie chart I use on your test actually has amount of time, and you calculate the percentages. This one here, so we have a linear graph. What I'm looking at is normal cells versus cancer cells. Notice the normal cells, cell number here. Normal cells are in blue. They have a lower cell count, right? And that makes sense because we know cancer is overgrown cells, out of control, unregulated cell growth. So these red lines, notice, are higher than the blue lines. So we would expect cancer cells to have a greater cell count than the normal cells. Here you have a graph, looks very similar to the graph you guys did in your math lab. So again, you should just be able to interpret a graph, compare. In this case, the normal cells are orange and the cancer cells are blue. So you, in this case, would indicate that there's more, a greater percent of time spent in interphase for the normal skin cells than the cancer cells, which we would expect because we know interphase goes too quickly. And then all of the rest, kind of the percentage in those other phases are longer. So really the big change here is interphase again. And then here is a data table. Looks very much like our math lab. In this case, the data table, you have to calculate the percent of total cells. So remember, you have to add up all of these cells together in the first place. Okay, so you got 153, here's 10. So 153, 163, 173, 175, 185, 195, 201. So there's 201 cells. So to calculate the percent of time spent in each of these phases, divide the number of cells found in that phase. So 153 divided by 201, and then times 100 to make it a percent. 26 divided by 201. Then multiply it by 100 to make it a percent. If you were calculating the amount of time, remember if you were given an amount of time that the cell spends in the whole cycle. So maybe it's a 24-hour cycle. So if we were calculating the amount of time metaphase spent, we'd say 12 divided by the total, 201. So 12 divided by 201 times the amount of time. So basically you're taking the percent and multiplying it by the amount the whole cell cycle takes. So 12 divided by 21 times 24. Okay, so there's example graphs and data tables and what you might have to do with them. So comparing cytokinesis and mitosis, how are they similar? They're both splitting something, right? How they're different is what's being split. Cyto, cell, or cytoplasm is splitting the cytoplasm or the cell itself. So here you see the cleavage furrow forming, that's cytokinesis. Mitosis is specifically splitting of the nucleus. So that had to do with the dance of the chromosomes, pairing up the sisters, lining them up at the equator, separating them, and then forming two new nuclei. That's mitosis, splitting of the nucleus in particular. So the spindle fibers, what do they do? What are they? They're microtubule filaments. So they're basically protein threads that are very small. And their job is to move the chromosomes around in cell division. So they'll connect to a chromosome, they'll move them to the center alignment, they shorten, they pull them apart, right? So their job is to move those chromosomes. If there was damage to those spindle fibers, they wouldn't be able to move the chromosomes correctly. And when the cell splits, the two nuclei may not be equal. They both may not have all of the contents that they're supposed to have. So they actually are pushing the cell further apart during the anaphase. We didn't really talk about that too much, but they push on the poles, and that's what's causing the elongation of the cell itself. We talked about stem cells, embryonic stem cells, and adult stem cells and how they're different. Both of them, first of all, their similarity, they're both have the potential, potential. They have the potential to become a variety of cells. Only the embryonic cell can become any type of cell. They're totipotent or pluripotent, which means they can become, the totipotent can become all of the cells of the body, and pluripotent can become most of the cells of the body. So they're going to form other stem cells that are found in the adults. The adult stem cells are multipotent or unipotent, right? So they've committed to a certain lineage, a certain line of cells. We talked about how these cells, once they are committed, they can only form cells of the inner part of the body, the organs, for example. These are going to be the middle part of the body, the bones, the muscles, the blood. These are going to be the outside of the body. So these are multipotent, and they've already chosen a line, so they're somewhat differentiated, but not entirely. So what are the advantages of using stem cells in medicine or in the lab? We can regrow types of cells, tissues, organs, use them for transplants. Adult stem cells are often taken from the individual themselves, and so you don't have to worry about a donor transplant rejection. This induced pluripotent stem cell, you read about that in the web quest. It's kind of cool where they take real cells and they kind of turn back time and make them stem cells. So we can use stem cells to treat all kinds of defects or damages, and we pointed out a bunch of these examples. Here's some common ones: diabetes, arthritis, brain injuries, and wound healing. Those are probably some of the more common ones. Cell differentiation refers to specialization of cells. So how this stem cell changes to be one of any kind of cell, to being one of only internal cells, to being an intestine cell. How this other cell differentiates, becomes specialized, exhibits only certain genes, to become one of those middle cells, to becoming bone. So when cells receive signals, certain genes are turned on, which makes them turn into a specific type of cell because they start exhibiting those genes and not other genes. And that's really what makes all of our cells different, is what genes are being expressed. So you have chromatin, chromatids, and chromosomes. You want to keep all of these words straight, right? Chromatin is the simplest form. In its thread-like version, chromatin is wrapped around histones, these proteins, during interphase, and it's going to continue to wrap. You can see the coiling here. Continues to coil round and round and round, and then it wraps back and forth upon itself, making it thicker and thicker and thicker until it's visible. Now we have what's called chromatid. So these are the duplicated DNAs. They're condensed chromatins, and they're used in reference to a single from a pair. So a pair is a chromosome, but the singles within the pair are called chromatids. So the chromosome is a pair of sisters, a sister chromatids. And when they separate, they no longer have that reference of being sisters. And so when they separate, they're once again called chromosomes. That part is kind of confusing. Apoptosis is referring to programmed cell death. So this is what's supposed to happen when cells cycle goes wrong. Like cancer cells are supposed to be rejected from the cell cycle, kicked out into G0, and then apoptosis occurs. Here you can see apoptosis. It's kind of like a bubbling and then a bursting of the cell itself. It's also what causes us to have like fingers and form. We start out as one big blob, right? Well, how does that blob turn from paddles to fingers? How does a tadpole lose its tail and become a frog? Apoptosis. So these cells that were once between our fingers, these cells that were once part of that tail, are programmed to basically decompose after a certain state of development. So they receive signals, or you have actually white blood cells that do this. They inject a chemical called perforin that causes a destruction of those cells. So we see it in our normal development, whether it's us or whether it's other animals. Even plants exhibit it when they drop their leaves. So apoptosis is programmed cell death. We talked about binary fission. This one wasn't on your review sheet, so I'm just going to review it. Mitosis does not occur in bacteria because mitosis is the splitting of a nucleus. So if it doesn't have a nucleus, bacteria, remember, are prokaryotic. If it doesn't have a nucleus, they can't divide. So bacteria have to do it a little bit differently. Remember, they have a single circular chromosome. They duplicate that single circular chromosome. Each chromosome gets attached to a different portion of the membrane. And then as the cell grows and elongates, there's a division of the cytoplasm. And then whatever cell membrane that chromosome is attached to is the new daughter cell that it becomes part of. So hopefully that's a good reminder of everything we talked about in the last 3 weeks. You should be all set for your test. Right now it's an Illuminate Ed, but I'm going to convert it into a Google form to make it easier for all of you who are there. Currently there are 36 questions. I know of at least one I'm going to drop because we didn't really do the cell surface to volume ratios. So maybe it's about 35 questions in the Google world, and I think you'll do great. So good luck to you. So proud of you guys for making it this far, kind of on your own, right? You guys need to be proud of yourself.